thank you in particular to Fabio for inviting me to present our experience here at CMDBuild Day. Yes, let me just present me a bit. I'm a computer scientist. I'm working at Istituto. Thank you so much to present the institution that it's a bit complicated to me, but just to briefly say that it's a veterinarian public institution. And for many activities, it works as a technical branch for the Italian Ministry of Health. We work a lot here as producing veterinarian information system from the Italian Ministry of Health. So we have a long experience on that, and of course also in the IT administration for servers, storages, and so on, in order to this application working. So as a computer scientist, I started here as a software developer, but also as a system administration. So as you can imagine, as a system administration, I know CMDBuild since a long time. Used it here, but at a given point in time, my career changes and goes to the bioinformatics areas, very interesting and also very quick evolving areas. And we were called to create the information system for microorganisms, for sequencing of microorganisms. So exactly the idea is to use CMDBuild. Now I explain how. I suppose that you can put my slides on. Yes, this is the CMDBuild for the management of genomic sequences of microorganisms. We use this in the National Official System and in several projects we have here at IZSAM. Next slide, please. Yes, the particularity of this presentation is not about IT configuration item, classical one, I mean, servers, storages, PCs, etc., or ticketing systems. But yes, our focus is on microorganisms. Next slide, please. And in particular on pathogens that typically are microorganisms found in patients or sick animals, and etc. Next, please. EndoDNA. It is a way on how microorganisms can be analyzed and understand and typed. Next slide, please. This is a way very well known in biology. I don't know how many poor IT people like me has knowledge about this, but in some sense, the laboratories for sequencing can be divided by arriving of microorganisms, DNA extraction, and the sequencing. The sequencing extract the DNA and read the DNA in order to transform the wet part, where organisms are, microorganisms are, to very likely for us, very nice bit and bytes in the dry part of the laboratories, where servers will analyze that and the storage are stored. Next slide, please. So using the NGS, the next generation sequencing, we now have a lot of results about organisms. That is called the raw data. That is the DNA readings exactly as it is, extracting from the sequencer, to the de novo assembly, and the typing in particular, the way how we can characterize these microorganisms. Let me say that this typing is something like if we see a human population, we would like to say, okay, this guy is blonde, this other is brown, blue eyes and brown eyes is characterization features of these microorganisms. Next slide, please. Additionally to the data from the genomes, we have to take in account the so-called metadata. That is what the species of the pathogens, the matrix, the source of isolation. This is an example. For instance, we could have viruses coming from the blood of a sheep, or maybe bacteria from chicken or from a sick patient. And then when and where? When has been taken and where has been collected this kind of microorganisms? It is important to understand the evolution of outbreaks. You know that we are living a very unfortunate days due to the COVID-19 crisis. So it's something that I think that you already have something. Next slide, please. Let me provide you this kind of example. Let us assume we have a number of pathogens. I just put as example these persons as it were pathogens. And we collected in some places all along Italy. Next slide, please. Well, one of use cases of data collection. Then we have the molecular typing. We try to characterize. Suppose that we have a method that is able to characterize these pathogens. For instance, we can, in this example, the method one is able to discriminate from men to women. Then we'll look for additional method to characterize more deeply with higher resolution characterize the pathogens. Next slide, please. The method two, we have a more accurate discrimination. We in example, divide blonde women from brown women. And next slide, please. And then we can calculate distance distances among these pathogens. Next slide. And we have the next slide, please. And we have the third use case where we can combine the typing of the pathogens together with the metadata where it's being calculated, focusing, underlying that we have a particular things, two pathogens that is man, are very close from typing. They are very several characteristics in common. Can be also understood as the same, but has been taken in a very distant places. This is something that should create an alert, because there is something that should be attentioned. Next slide, please. That is exactly what we have in a dashboard. We collect, you can see on the right, great numbers of pathogens, where we select a subtree of pathogens. The more are close, the more are similar in the pathogens, and on the right, combined with the day where they have been taken, saying that there is something that is being flowed from one point to another. Next slide. Using next generation sequencing and epidemiological activities like the one illustrator, there has been proved to be the possibility to have a higher level of outbreaks discovered. And as you can see from the yellow bars, at the contrary, we have less people involved, less people sick. So we are able to find more outbreaks and to have less person sick. Next slide, please. Okay, so we arrived, that is things that are well known all over the world. In the 2017, we formalized the need to have a National Reference Center for WGS, whole genome sequencing of microbial pathogens in Italy, and we be nominated by the Italian Minister of Health on this on 2017. In my idea, it was immediately thought to have, for the database and the analysis storage, to use a system like the CMDBuild. Next slide, please. The system, as you can see, is a classical, we can say, web application where we can store two kinds of very different things. Metadata, which is very close to the classical relation data we use in other information system, together with this typing data coming from the pathogens and analysis of several kind, because there is a very fast growing of new methods we can apply to pathogens in order to be more precise and more correct to discriminate pathogens, and provide back some of the analysis I've illustrated. Next slide, please. Yes, this is just a screenshot of the home page, which is a classical page from CMDBuild. Next slide, please. This is the architecture, the very particular architecture, because we had the need in the GEMPAT system, the orange one in the center, to take data coming from different organizations that he was the owner of the pathogens data. We have, for instance, the Listeria, Brucella, and the Campylobacter, which has three kinds of pathogens coming from the relative national reference center and national reference laboratories, are together bring in the GEMPAT system. It was an important part, the one related to harmonization of those data, and to harmonization of also the methods, because we might have different methods for different pathogens. I have put cheese, chicken, and cows just to describe that these pathogens are, which is the main matrix that is the host where can be taken this kind of pathogens. Listeria is common in cheese, Campylobacter in chicken, and Brucella in cows. Moreover, at this, you see in the yellow are metadata and the blue one are the results of the WGS analysis, aiming on type, the microorganisms. On the yellow boxes on the left, we have the national information system, official national information system, like the animal holding and movements, very useful for the cows. So for Brucella, the CMAN, the animal outbreak notification, it is important to understand if we have holdings that are present animal 6, and CINSA, which is a system for food companies. All these are official national systems that are brought together in GEMPAT in order to improve the metadata of this kind of pathogens. All these things are in order to provide means for outbreak investigations and surveillance. Next slide, please. It is important to mention which of the part has been involved of CMDBuild for this kind of things. In particular, the great part we have needed for this is the web service and connectors, as you can imagine, because we have a lot of information system to integrate and to harmonize. But of course, the history system has been very, very useful, as also the report engine. But the great part and the main reason we use CMDBuild for this kind of information system is its high flexibilities in order to add characteristics, information whenever a new pathogen arrives, having the need to register different information, So the high flexibility of CMDBuild is the main important features we use for these things. Next slide, please. Yes, we have some report, as you can imagine, dating from the report. Next slide. This is the features I mentioned in the example of peoples. This is the pages where we can filter for samples and then require an analysis for the typing analysis of the pathogen we selected. And then, next slide, please. We can have the typing report, like in this way, when we can see, the orange one and the light orange are samples coming from humans because we have blood or liquor, while on the blue one we have prosciutto, which we can understand there is human cases very close to meat products. It's very important. They should trigger an alarm, an alert. Next slide, please. And of course, we have the similar dashboard that I mentioned at the beginning, where we can have a subset of very close microorganisms taken from different places, different regions in this case. That is, again, another trigger that should be, another alert that should be triggered. Next slide, please. Okay, given the success of this national official information system, we use the same ideas in order to provide a system in this One Health AGP program. It is a European program aiming to deal with the One Health. One word, two words on One Health. One Health means to study human sickness, the patients, together with the animal and together with the environment, because all this information together leads to a better surveillance, so to reduce the number of sick patients. So it is dealing with the needs to combine information system, just to translate to our IT people, to combine information system coming from different organizations, from the public health to the veterinarian environment. Next slide, please. In this place, as you can see, is very similar architecture. We provide this kind of prototype information system that one member state can implement. Indeed, at the end of the project, which is next year, this cohesive information system will be released open source on GitHub. And as you can see, one member state can use it in order to combine information from the public health pathogens databases, let me say very simplifying, on the left top, together with the veterinarian's information systems on the right, together in one system, again improving the metadata with the left box, yellow boxes, in order to provide outbreak investigation, surveillance, and of course, reporting to the European level agencies. Next slide, please. These are we provided 3 demos of the system for Italy, Norway, and Holland, that are the member states involved in the so-called feasibility studies. So we provide a prototype together with CMDBuild, for this as a playground, we can say. It's not official, just a prototype, where the member states can use in order to understand how the integration between data can be realized. Next slide, please. Another important project is this one, because at the Italian level, we are also working, we as a veterinarian institution, together with the public health institution, as you can see in Italy is the Istituto Superiore di Sanità, with integrating the 2 information systems related to pathogens from a human side at the Istituto Superiore di Sanità, data coming from the hospitals, together with the veterinarian part at GEMPAT level, coming from the holdings and the products and the supermarket and so on, in order to have an integrated system. And you can imagine that it's been designed by me and the counterpart of the Istituto using CMDBuild. We are in the prototype phase, and we are. It is foreseen that by the end of this year, first month of next year, we will present it to the Italian Ministry of Health. Next slide, please. Again, it seems that the idea is good, and also other people like this kind of ideas. So indeed, the Norway Veterinary Institute asked for the possibility to integrate a CMDBuild in a GEMPAT version for microorganisms in their architecture internal to manage pathogen at an NBI. Next slide, please. So at the end of the presentation is just to say there is a high number of projects where we use CMDBuild, and I would like again to underline the highly flexibility of the system. That is the main part that allowed us to integrate the GenPath system together with other national official information systems. Thank you very much.